Hey everyone, in this presentation I'm going to be teaching everyone about CRISPR, which has changed the game for sickle cell disease patients and has been able to correct the sickle cell disease phenotype. The main research paper used in this presentation was editing a gamma globin repressor binding site restores fetal hemoglobin synthesis and corrects the sickle cell disease phenotype. So before I dive into how sickle cell disease can be treated, I first need to cover what sickle cell disease exactly is. Sickle cell disease is an autosomal recessive disease in which red blood cells deform into a sickle shape. The Punnett square on this slide shows that if two parents are heterozygous for sickle cell disease, for each child they have, there is a 25% chance that they will have sickle cell disease. Why is the sickling of red blood cells so significant, you may ask? The red blood cells in our body are especially important as they carry oxygen to all parts of the body. For a person with sickle cell disease, their red blood cells become hard and sticky and begin to look crescent-shaped. The image in the bottom right of the slide portrays how red blood cells should work and how red blood cells work inside an individual with sickle cell disease. The top of the image shows the normal flow of the red blood cells throughout the blood vessel. In contrast, the bottom of the image shows how sickled red blood cells can stick to the side of the blood vessels and block healthy red blood cells from transporting oxygen to the body's tissues and organs, which can cause infections, acute chest syndrome, and even a stroke. When the body lacks enough red blood cells to carry enough oxygen, it is called anemia, or also known as low hemoglobin, which can make you feel tired and weak. Hemoglobin is a protein in your red blood cells that helps carry oxygen to your body's organs and tissues. Fetal hemoglobin, also known as HbF, is found in fetal red blood cells and is involved in transporting oxygen from the mother's bloodstream to organs and tissues in the fetus, while adult hemoglobin, aka hemoglobin A, makes up 95 to 98% of adult hemoglobin. The structure of both of these hemoglobins is quite similar as they both have two identical alpha subunits, which is shown on the top of the diagram. But in fetal hemoglobin, the two subunits are gamma, while in adult hemoglobin, they are beta, as shown in the image. It should be noted that fetal hemoglobin has a very high affinity for oxygen. If you look at the graph, you can notice that the percent oxygen saturation is much higher in fetal hemoglobin and reaches 100%, while adult hemoglobin reaches only about 95%. Okay, so what exactly happens in your body to cause this sickling effect? Beta hemoglobinopathies are severe anemias characterized by abnormal or reduced amounts of hemoglobin beta chains. Sickle cell disease and beta thalassemia are by far the most common monogenic disorders, as people who inherit beta thalassemia produce very low levels of hemoglobin, which in turn leads to a lack of oxygen in many parts of the body. Affected individuals also have a shortage of red blood cells, which can cause pale skin, weakness, and complications. People who inherit sickle cell anemia obtain abnormal hemoglobin called sickle hemoglobin, also known as HbS. Sickle hemoglobin is the result of a point mutation at the 6th amino acid in the beta globin chain, as shown in the picture. Because of this mutation, the translation is affected, which in turn leads to a different amino acid being coded for. Because of this mutation, the valine type hemoglobin causes red blood cells to sickle. The management of sickle cell disease is primarily focused on avoiding pain episodes in the first place, while also relieving symptoms. One way of treating sickle cell disease is by taking medications. For example, L-glutamine oral powder, which is a drug that reduces the frequency of pain crisis treatments for individuals with sickle cell disease. Moreover, another example is hydroxyurea, which also reduces the frequency of painful crises and may also reduce the need for blood transfusions and hospitalizations. Talking about blood transfusions, the picture on the bottom right of the slide is an image of a sickle cell disease patient during a blood transfusion. Blood transfusion is the process of providing normal red blood cells to the patient's body. Red blood cell transfusions help lessen anemia while also reducing the blood's viscosity, which prevents complications. Another treatment option is stem cell transplants, but it's best to steer away from them because there are numerous risks involved with this procedure, including death. Needless to say, the bill is also very hefty, ranging from a whopping $350,000 to $800,000. Although these treatments can be quite beneficial, there are a significant amount of risks involved with them. But wait, there's more. And by more, I mean CRISPR-Cas9, which is short for clustered regularly interspaced short palindromic repeats, which is a mouthful, so let's just refer to it as CRISPR. But what is so special about CRISPR, you may ask? And that's that CRISPR is a molecular tool that scientists can utilize to accurately target and cut any kind of genetic material you could possibly think of, which is why the silly graphic on the side is actually very accurate, as CRISPR-Cas9 hunts down a specific DNA sequence and modifies it or deletes it. Similar to searching for a word in a Google Doc and deleting a letter or adding one. Furthermore, CRISPR is the easiest and most convenient method scientists have ever had to manipulate the DNA of any organism. CRISPR consists of two components, the Cas9 protein that cuts the DNA, and the guide RNA, aka the gRNA, which recognizes the sequence of DNA that needs to be edited. Once the scientists realize which sequence they want to cut out, they insert the CRISPR-Cas9 complex into the cell and can then edit the DNA or delete it as they wish. In a clinical setting, scientists discovered that some patients, despite having sickle cell disease, did not manifest the symptoms of a typical sickle cell disease patient. This tickled the scientists' brain and they decided to run lab work. The results portray that these patients produced an abnormally high amount of fetal hemoglobin, which was very odd because fetal hemoglobin is turned off right after birth. They later found out this was a condition known as hereditary persistence of fetal hemoglobin, which is a result of mutations leading to the lack of inhibitory protein production. The scientists then decided to focus on several areas of interest within the mutated DNA to find out which genes are responsible for this. They discovered that the areas on chromosome 11 responsible for the production of fetal hemoglobin were HBG1 and HBG2. On the other hand, the beta chains of adult hemoglobin are produced by HBB. Within HBG1 and HBG2, they identified 5 areas of genetic sequence which they named -197, -196, -195, -158, and -115. All 5 of these points are responsible for making repressors called LRF and BC11A. These two repressors convey signals to HBG1 and HBG2 to shut down gamma globin production at the time of birth. Obviously, for the patients who produce fetal hemoglobin, the repressors were never made, therefore the production of fetal hemoglobin were never turned off. The drawing on the left was made by me to explain how fetal hemoglobin should be stopped at birth. Knowing the points of mutation now, the scientists turned to CRISPR to mimic the exact same mutation for a patient without hereditary persistence of fetal hemoglobin in an attempt to ease their symptoms. To make what I just said sound a little more understandable, the scientists basically realized, oh, well, if fetal hemoglobin helps ease symptoms, we can manually do it by cutting out the code that represses fetal hemoglobin by using CRISPR. The scientists then identified the 5 areas of interest and used CRISPR to remove the sequences responsible for making the LRF and BC11A repressor proteins. As a result, every single intentional mutation led to a varying degree of increased production of gamma globin, which in turn resulted in high levels of persistent fetal hemoglobin, which means that the experiment was a success. The next few slides elaborate the data points analyzed and the results gauged against the control. The scientists ran a PCR test on all 5 areas of interest and compared the mutations to the control, which you can think of as a patient who has sickle cell disease and produces no fetal hemoglobin. The bar graph may look intimidating, but think of it as the dark blue is bad and the light blue is good. This is because light blue means that there is fetal hemoglobin being produced. All 5 points had varying degrees of success, with each mutation having around 40% fetal hemoglobin, while AAVS1 barely had 5% of fetal hemoglobin. Flow cytometry plots were also used to show the data. In this case, the closer the points are to the right, the greater the fetal hemoglobin levels are, clearly showing that AAVS1 had little to no fetal hemoglobin and that the 5 points had a great amount of fetal hemoglobin. Another way of determining the success of the experiment was by looking at the red blood cells under a microscope, and clearly there were little to no sickle cells in the 5 sites. For the bar graph, all 5 points had a great amount of non-sickle cells and once again showed success. In conclusion, the use of CRISPR shows promising results and will most definitely be the future of medicine. Since the publication of this article was in February of 2020, there have been numerous successful experiments using CRISPR techniques in mammals with promising results recently. Researchers are hopeful that with new research and cutting-edge technology, they will be able to create more successful changes to specific parts of DNA and cure illnesses such as malaria, HIV, cancer, etc. Moreover, CRISPR technology also bears the potential to revive extinct species in the future and can even create completely new species, which brings us to the next topic. Of course, with good, there is also bad. In the case of CRISPR, that would be an ongoing ethical debate about messing around with DNA. There are a few main discussed problems, such as the risk of uncertainty of the technology and its application. Since, after all, CRISPR is still considered a newly founded technology, and we are learning more about it each day. The second issue is whether or not scientists should use CRISPR in embryos to prevent diseases such as Huntington's, cystic fibrosis, etc. This is a burning question where many scientists are split whether or not this is the right thing to do. I want to thank everyone for listening to my presentation, and I hope everyone has learned something that they can apply to their life. And I also hope everybody has a good summer. Here is my works cited page with all the sources I used throughout my presentation.